everyone. Today I'd like to introduce our recent study that named as Metagenomic sequencing combined with flow cytometry facilitates the absolute quantification and accurate microbial risk assessment of broad-range bacterial pathogens in municipal wastewater without cultivation. Metagenomic wastewater monitoring records the dynamics of bacterial pathogens in urban and livestock wastewater as an early warning tool for viral detection in local communities. However, microbial kinetics measured by current standard microbiome methods is limited by the nature of microbial composition. At present, three absolute quantification methods have been developed. The first approach relies on a qPCR assay of a broad-range 16S rRNA gene to estimate the total bacterial load, followed by amplicon sequencing to obtain the relative abundance of the bacterial community. The second approach requires the spike-in of known absolute abundance of a given pathogen for calibration. The third approach involves the use of direct measurement of the total bacterial load by flow cytometry. Based on the above three methods, we first established a workflow that combining metagenomic sequencing and flow cytometry for risk assessment of three mock samples. In this study, three simulated communities consist of 2, 8, and 32 bacterial species were established to evaluate the best informatic tools for classification analysis, with total assembly size range from 8.2 to 130 megabytes. We then determined the DNA extraction efficiency of 128 bacterial species. DNA extraction efficiency ranged from 9.6 to 70.8%. Gram-negative bacteria also showed significantly higher DNA recovery efficiency than gram-positive bacteria. Then the DNA extraction efficiency of individual bacterial species were also used to correct the original RA data. We then evaluated the accuracy and boundary of inferred concentrations. Corrected RA data has the lowest RMSE value. Inferred concentrations also showed a good linear relationship with expected value. The result shows that 94.5% of IC errors were identified from the data which RA below 5%, suggesting that species with low abundance were the major sources of the IC errors. The variance in the relationship with expected concentration tended to be inversely proportional to species concentrations. We also evaluated the real sample using this workflow. The Yalu River forms the northwest border between North Korea and China. From May 5 to September 5, 2021, wastewater samples were collected to monitor the dynamics of pathogens for case studies. The sampling site is located at a municipal wastewater discharge point in Dandong City. Samples were collected fortnightly. We also evaluated the resolution of Metagenome-assembled genomes for identification of virulence and antibiotic-resistant genes. Binding showed a good quality of MAX from real samples. The relative abundance and N50 value of MAX were obtained from metagenomic dataset in wastewater. In summary, we firstly developed a workflow combining metagenomic sequencing and flow cytometry. We then evaluated its robustness for the absolute quantification and microbial risk assessment of specific pathogens in both mock and real samples. Next, results show that this workflow accurately estimated the absolute abundance of pathogens in mock communities and real samples. Last but not least, genome information extracted from MAX has further led to the microbial risk assessment of bacterial virulence and antibiotic-resistant gene profiles, which facilitates the identification of novel virulent strains in the challenge settings.